Hello friends, in this video I'm going to take the detail of glycine metabolism, its uses and the disorders also here in glycine. So let's get started. Glycine, the simplest, smallest amino acid you know, and this I already told you, it is non-essential. We can easily make this simple glycine in our body, so we don't require it in diet. We can make it in our body, so it is non-essential. And regarding the polarity of glycine, I told you it is a controversy. Some books say it is polar, some books say it is non-polar. So according to the question, you will solve this thing, right? You just note it down. It is a controversy for glycine, right? And one more thing, that glycine is glucogenic amino acid, that is, it can form glucose, right? Now, glycine is synthesized from carbon dioxide and amino group. They join to form glycine, and this is enzyme involved here is glycine synthase. Glycine synthase enzyme. It is a multi-enzyme complex which requires vitamin B6, vitamin B9, that is tetrahydrofolate, and it also requires lipoic acid and NAD, right? So here NAD is required, vitamin B6 is required, lipoic acid is required. It is a multi-enzyme complex. Glycine synthase requires so many coenzymes. Like I told you, link reaction enzyme pyruvate dehydrogenase is a multi-enzyme complex, right? Here N5N10 methyl tetrahydrofolate will be giving methyl group, making tetrahydrofolate. Now glycine is having 2 carbon. Glycine can be again broken down to carbon dioxide and ammonia by glycine cleavage system, and now methyl group is removed, and again tetrahydrofolate will be converted to N5N10 methyl tetrahydrofolate. This is done by glycine cleavage system. Right now, glycine is converted to serine. This reaction is reversible and it occurs with enzyme serine hydroxymethyl transferase. Serine hydroxymethyl transferase. Here also N5N10 methyl tetrahydrofolate is giving carbon, so that serine is having 3 carbons, right? This serine will then further form pyruvate. So as glycine and serine in the catabolism end, they are forming pyruvate. So I will say glycine and serine both are glucogenic amino acids. You know, any amino acid which can form pyruvate, that is glucogenic, right? So in the synthesis of glycine, one more thing is there, threonine. It can also be formed from threonine by the enzyme threonine aldolase enzyme required here. So you can note one point here, that glycine metabolism is very much related to B6 and B9, that is B9 is tetrahydrofolate. So here also tetrahydrofolate is required. So glycine metabolism is very much linked with tetrahydrofolate and vitamin B6 metabolism, right? Now glycine gets converted to glyoxylate, and here enzyme is glycine oxidase. Glyoxylate also is 2 carbon. Like glycine, it is also 2 carbon. Glyoxylate can be converted to formate by removal of 1 carbon dioxide. You know, formate is HCOOH. It is having only 1 carbon. On the other hand, glyoxylate on oxidation will be converted to oxalate. Oxalate is having 2 carbons, 2 carboxy group here like this, and oxalate can then be excreted in urine, right? Now glyoxylate can be again converted to glycine, and here the enzyme required is glycine alanine transaminase. If this enzyme is deficient, that is glyoxylate cannot be converted to glycine. Along with this enzyme deficiency, there is a defect of glyoxylate to formate conversion. Then you can see here that excess of glyoxylate will be forming excess of oxalate, and this is known as primary hyperoxaluria. Primary hyperoxaluria is actually a protein targeting disorder. It is a protein targeting disorder these patients have oxalate stones in kidneys, oxalate stones in kidneys. There is also oxalate deposition in extrarenal tissues, also in kidneys, also in extrarenal tissues. And here the treatment given is again hydration given to prevent the formation of these oxalate stones, and there is restriction of oxalate rich food, example green leafy vegetables, beetroot and tea, etc. Now this was the case of primary hyperoxaluria. Now let's see what is secondary hyperoxaluria. Secondary hyperoxaluria due to some secondary causes. Here vitamin B6 deficiency. In the deficiency of vitamin B6, glycine metabolism will not occur and excess of glycine will be forming excess of oxalate. So vitamin B6 deficiency is one reason. Vitamin C toxicity is another reason. Vitamin C toxicity. How vitamin C toxicity is leading to hyperoxaluria? You know, vitamin C is ascorbic acid. In the metabolism of ascorbic acid, dehydroascorbic acid is formed, and dehydroascorbic acid is converted to oxalic acid. If excess of vitamin C, then excess of oxalic acid will be formed, leading to secondary hyperoxaluria. Also, ethylene glycol poisoning will lead to this condition. What happens in ethylene glycol poisoning? Glycolic acid is the main toxic compound which is formed in this poisoning. This glycolic acid is converted to glyoxylate, and glyoxylate, you know, will form oxalates, and these oxalates can cause kidney damage. So they say that ethylene glycol poisoning can cause kidney damage. And one more reason you can write, bariatric surgery. Bariatric surgery will also lead to secondary hyperoxaluria, right? Then coming to other disorder, that is non-ketotic hyperglycinemia. Non-ketotic hyperglycinemia. Here in these patients, there is a defect in glycine cleavage system. Defect in glycine cleavage system, which is the major pathway of glycine catabolism. Now, when glycine catabolism is not occurring, there is increased glycine. You know, glycine is a neurotransmitter. Symptoms will be appearing here. Clinical features of these patients. These patients are mentally retarded. They have seizures, lethargy, apnea, increased glycine, abnormal high level of glycine found in blood, CSF, and also in urine. This is also known as glycine encephalopathy. And why it is called non-ketotic hyperglycinemia? Because there are many other inborn errors of metabolism where there is ketotic hyperglycinemia. One example is propionic acidemia, where ketotic hyperglycinemia occurs, right? But this one is non-ketotic hyperglycinemia. Ketone bodies are not increased in the blood of these patients, and there is no effective treatment of this disease, right? Now coming to another disorder of glycine metabolism, that is glycinuria. Glycinuria is defective reabsorption of two amino acids. Defective reabsorption of two amino acids. One, you know, as the name tells you, glycine, and one more is proline. Actually, the transporter of glycine and proline is common. That transporter is defective here. So both amino acids are not properly reabsorbed. So in the urine of these patients, there will be glycine and proline, right? So here you see, but in these patients, serum glycine levels are normal. Urine glycine levels are increased, but serum glycine levels are normal. That's why it is not glycinemia, it is glycinuria. These patients also have increased risk of oxalate stones. Increased risk of oxalate stones but urine oxalate is normal. Urine oxalate is normal, but they have increased risk of oxalate stones. Now coming to uses of glycine. Uses of glycine. Glycine is used for the formation of heme, for the formation of reducing agent, glutathione. It is also used in the formation of purine rings, but it is not required for pyrimidines. And also glycine is used for the formation of serine, and glycine is also a conjugating agent, like it is conjugating bile acids. I also told you it is a neurotransmitter. It is a neurotransmitter, excitatory as well as inhibitory. And also glycine will form creatinine. And there's one more role of glycine, that is formation of choline. These two compound synthesis I'm going to take in detail. So first I'm going to take the synthesis of creatinine, right? Now coming to the detail of formation of creatinine. Here the starting material is one amino acid, basic amino acid, that is arginine. Arginine combines with another amino acid, that is glycine. They both join to form guanidinoacetate. Guanidinoacetate, and this reaction is occurring in kidneys. Guanidinoacetate will be then converted to creatine. In the formation of creatine from guanidinoacetate, methyl group is required. This reaction is occurring in liver, and the donor of this methyl group is SAM, S-adenosylmethionine, taken for giving methyl group gets converted to S-adenosyl homocysteine. This reaction occurring in liver. Now creatine is from liver, creatine will be going into blood, and from blood this creatine will be taken up into the muscle. Let's say this is muscle cell. Here creatine is there in muscles. Now this creatine will be converted to phosphocreatine. Creatine converted to phosphocreatine, and here in this reaction ATP will be used to add this phosphate group, and phosphocreatine will be then converted to creatinine. When phosphocreatine is converted to creatinine, here there is formation of ATP. This phosphate group can be taken by ADP and ATP is formed. As ATP is formed in this reaction, so we say that phosphocreatine is the immediate source of ATP for muscles. This is also a frequently asked question. Immediate source of ATP for muscles is this phosphocreatine. And also creatine can also form creatinine directly without the formation of phosphocreatine, but more creatinine is formed from phosphocreatine as compared to creatine. Now this creatinine is just a waste product of metabolism of creatine, and it is thrown out via kidneys. This in blood also creatine can be converted to creatinine, that is also thrown out of the body via kidneys, right? So this way creatinine is formed from glycine amino acid, and one more amino acid which is required here is arginine is also required here, right? Now next is we have to see the formation of choline from glycine. Choline is required for the formation of phospholipids, right? So this is very important thing in the body. Let's see how glycine is converted to choline. Glycine, you know this saying that glycine will form serine. Serine is a 3-carbon compound, glycine is a 2-carbon compound. Now serine will be decarboxylated. When serine is decarboxylated, then ethanolamine is formed. You know that from amino acid, if acid group is removed, we are left with amine, and the name here is ethanolamine. Now ethanolamine is 2-carbon because 1 carbon is removed here, right? Now ethanolamine will be converted to, choline. Choline is nothing but trimethyl-ethanolamine. Trimethyl-ethanolamine, that is here 3 methyl groups are added with the help of SAM, that is S-adenosylmethionine. Now trimethyl-ethanolamine or choline will be converted to betaine, and here the enzyme is choline dehydrogenase. Enzyme is choline dehydrogenase, and this betaine is also called trimethyl glycine. Choline is trimethyl ethanolamine, and this betaine is trimethyl glycine. Now this from trimethyl glycine, when we will remove one methyl group, then we get dimethyl glycine. From dimethyl glycine also one more methyl group is removed and we get N-methyl glycine, which is also known as sarcosine. From sarcosine one more methyl group is removed and we get glycine. So sarcosine is N-methyl glycine. These all MCQs are asked. Betaine is trimethyl glycine. Choline is trimethyl ethanolamine. And serine on decarboxylation will give ethanolamine, right? So here in this metabolism, on this side methyl group is getting added, on this side methyl group is getting removed. These all contribute to one carbon pool of the body, that here one carbon pool is giving methyl groups, and these methyl groups are again entering into one carbon pool, and mainly folate is responsible for one carbon pool. So I will say that glycine metabolism is very much interlinked with folate metabolism, right? Also choline and betaine metabolism is also linked to tetrahydrofolate metabolism, right? This is it with glycine metabolism. Best wishes to you.